Hello students, welcome to SES channel. In the last class, we have discussed about the chlorophyll and how essential it is for photosynthesis. We have taken an experiment. In experiment, we have chosen a plant with variegated leaves. Variegated leaves like any money plant or crotons, where few parts of the leaf are green in color. That is, the part of the leaf which is green in color contains chlorophyll, and the other part of the leaf which is not in green in color, it doesn't have chlorophyll. So the part which has chlorophyll prepares food. That food is given to the other part. Got it, children? For example, here, this is the variegated leaf. This green part prepares food. Okay, starch is prepared over here. It contains chlorophyll. This part, which is somewhat yellowish in color, it doesn't have chlorophyll. This chlorophyll, this food is provided to this part. That is why we have chosen this variegated leaf. Okay, to show the presence of chlorophyll. Now we'll discuss in detail about this chlorophyll, how the photosynthesis occurs in green plant. So if you look at the picture of this, where is chlorophyll present? Chlorophyll is present in chloroplast. Chloroplast. Already I have shown you the cross section of the leaf. See here, this is mesophyll, right? This is spongy mesophyll. This is palisade mesophyll here, and here you have the dots, green dots. That is the chloroplast, isn't it? In the chloroplast, look at the structure of this chloroplast. This is the outer membrane and this is the inner membrane. Outer membrane will differentiate this chloroplast from other cell organelles. Okay, then in the inner membrane, inside this, you can see oval shaped disc-like structures, isn't it? These are called thylakoids. These are called thylakoids. They are piled one upon the other to form a structure called granum. To form a structure called granum, plural it is grana. So this is the stroma or it is matrix. That fluid-like substance, it is the stroma, and this is the connecting stroma lamellae. Okay, this is important for photosynthesis. The chlorophyll present in the thylakoid, it absorbs light. It absorbs light. We'll see that process in steps. See, light enters the chloroplast of the leaf cells. Then the chlorophyll, as I told you, chlorophyll is present in the thylakoid, that is present in the grana, and it captures the light energy. It captures the light energy. By today, energy ni capture cheskoni. Already we have seen the events of photosynthesis. First process enti absorption of the light energy and conversion of that light energy into chemical energy. Yes, that energy is absorbed by the green pigment present in grana or in the thylakoids, that is chlorophyll. So a part of energy it is used. The next event is water molecule is split into hydrogen and oxygen. I told you, splitting of water molecule. So part of energy which is utilized, which is absorbed by the chlorophyll, that is used in splitting of water molecule into which the water. How do you get water? How these green plants get water? Yes, through the xylem. Yes, through xylem. So that is split into hydrogen ions and oxygen. The other part of the energy that is used, that is sent to stroma. That is sent to stroma. Okay, that is already I have described about the stroma. So what will happen again? This oxygen is the byproduct. This oxygen is the byproduct of photosynthesis. Already we have discussed that. That is released into air. From here the oxygen goes out into air. Then hydrogen, that is carried for further preparation of food. Hydrogen is carried for further preparation of food, that is sent into stroma. Okay, so the photosynthesis process, actually the conversion, that is the reduction process. Now the carbon dioxide in the stroma, you know about the stomata, how the gaseous exchange takes place and the carbon dioxide enters into the stomata. Then this will receive the carbon dioxide, and here in the stoma, this carbon dioxide and hydrogen ions also enter here. They combine to form carbohydrates. So they combine to form carbohydrates. So hydrogen, this water molecules and carbon dioxide, they combine to form carbohydrate. Here it is glucose. So the glucose which is formed here, it is sent to different parts of the leaf for the growth or for different functions of the plant. So different parts of the plant. And any carbohydrate which is more than what is required, that is stored in the form of starch. That is stored in the form of starch. First lo cheppanu, mana human beings lo aithe excess of whatever nutrition or food it is prepared, that is stored in the form of glycogen. In plants it is stored in the form of starch. Got it? This is the process of photosynthesis that occurs in green plants. I have explained this in detail. So what happens? The light enters into the chloroplast. That is a figure of chloroplast. I have explained that. Chlorophyll present in the grana, it captures or absorbs the light from the environment, and part of the energy it is used to split the water molecule so that hydrogen and oxygen are released. Okay, the oxygen is a byproduct and that is released into air. Hydrogen, it is sent into stroma where hydrogen combines with carbon dioxide and water, and what is formed? Glucose. Glucose is formed. Glucose is broken down again, and it gives energy. It gives energy to different parts of the plant to perform different functions, and the rest of the glucose which is not utilized, it is stored in the form of starch. So what are the functions of thylakoid and stroma? You should know this mainly. Thylakoids, I told you these are the oval-like structures which pile up to form granum. Thylakoids, they help in water photolysis. Photolysis means break. The water is split into hydrogen ions and oxygen. Okay. Then it helps in electron transport change and ATP synthesis. ATP is adenosine triphosphate. Energy we measure in ATP, isn't it? ATP is adenosine triphosphate. So adenosine triphosphate, it is the enzyme which is essential for ATP synthesis over here, and oxygen is the produced here in thylakoids. Already have shown that oxygen is produced as a byproduct. Coming to stroma, it is matrix, it is fluid-like substance. It contains enzymes, ribosomes and DNA. Glucose production, actual photosynthesis happens here, that is carbon dioxide is reduced to carbohydrates over here. So the actual Kelvin cycle which is used for this carbon dioxide which is used here, this is used in Kelvin cycle. You know, you'll get that Kelvin cycle in higher classes where carbon is fixed, carbon fixation, and later the further process ATP, utilizing ATP energy in the form of ATP and ADPH, utilizing these energies. Again, these are converted to form complex substances. Okay. Now coming to the next raw material or element which is essential in photosynthesis of it is carbon dioxide. First we have covered chlorophyll. Next it is Carbon dioxide. So from where do the plants obtain carbon dioxide? Look at the picture. Already have shown the cross section of leaf. Underneath the leaf, underside of the leaf, you can find these structures, isn't it? These structures. This is like a kidney shape or bean shape, isn't it? These are called guard cells. This aperture or a hole, or this is called stoma. This is called stomatal pore. This is called stoma or stomatal pore. So this stoma, this is aperture, it is guarded by this guard cell. Whenever water enters through this, whenever you can see the green dots over here too, yeah, those are chloroplasts. Whenever water enters into this guard cells, what will happen? Yes, it will become turgid, that is swollen. It will become turgid or swollen, and the inner walls of this guard cells, they are pulled apart so that it opens. So whenever water is entered, water molecules enter here, they become swollen or turgid, and the inner walls of this guard cells are pulled apart so that there is an opening which will let the atmospheric carbon dioxide to enter in. Got it? Got it. When the water moves out, when water is moving out, what will happen? The guard cells are they are shrunken here, that is they shrink and they will close the stoma. So that no gases can move. Okay, this is how the gaseous exchange takes place. Got it? Opening of this aperture and closing of this will help in gaseous exchange. Coming to the photosynthesis process, I told you carbon dioxide is taken in and oxygen is discarded. That is the byproduct. Photosynthesis. While in respiration, opposite happens. Carbon dioxide is discarded and oxygen is taken out. So this process is through the gaseous exchange. This occurs in spongy mesophyll where stomata are present. If you look at this figure, stomata, some epidermal cells, they undergo some specialization in their shape or in their size, and they form subsidiary cells, and this is called stomatal apparatus. This is called altogether the guard cells, the stoma and this subsidiary cells. Altogether it is called stomatal apparatus. Okay, this is the explanation what I have given. If you have any doubts, please comment below. And if you didn't subscribe the channel, like it and share to as many friends as possible. Stoma is singular, stomata is plural, children. That's all. Stomata, it is not only present in the leaf, it is also present in the surface of stems, roots and leaves for gaseous exchange. So the gaseous exchange also occurs in stems, roots and leaves. Coming to during daytime, what is going to happen during daytime? The simple process already I have told you. The exchange of this goes through by the simple process. What is going to happen with how is the water taken up? Simple process, osmosis. Through osmosis during daytime, water is taken up and the guard cells, they become turgid. When they become turgid, the inner walls are pulled apart so that it opens, the stoma will open and carbon dioxide enters. Got it? Got it. Whereas in night, when water is lost, these will close, the inner walls will close. How? Because that is shrink. The guard cells are shrunken, and what will happen? The apertures or the stoma will close. Okay? Got it? Now, coming to the experiment. This is an experiment. As I told you earlier, activities are very important for you because these can be asked for 8-mark question. Now, an experiment is conducted to say that carbon dioxide is necessary for photosynthesis. This is a simple experiment. The common things what we have done is we'll take 2 plants there in the chlorophyll experiment. That was only 1 plant, and we have chosen a plant with variegated leaves. What are variegated leaves? I have explained. Now we'll choose 2 plants, similar plants will take up, same plants, we'll keep that plants in dark room for 3 days. The same thing. Keep them in dark room for same size and same plant will take and keep them in dark room for 3 days. Now place each plant in glass plates. You place each plant in glass plate, okay. Now seal this with any Vaseline or anything so that vacuum is created and air doesn't enter. Carbon dioxide is air, isn't it? It should not enter. So you seal it with some Vaseline so that vacuum is created and air doesn't enter. Now place a watch glass containing potassium hydroxide. You can observe in near the pot X గమనిస్తున్నారా, అక్కడ ఒక ప్లేట్ ఉంది, గ్లాస్ ప్లేట్, దాంట్లో potassium hydroxide. You are placing potassium hydroxide. Okay. Now cover the both plants with bell jars. This is also called as bell jar experiment. Cover this both plants with bell jars. Use Vaseline, as I told you, to seal the bottom of the jar so that it becomes airtight. Now keep this plant in sunlight for 2 hours. Pluck a leaf on it. Pluck a leaf from each plant. After 2 hours, pluck a leaf from each plant. Do both the plants show the same amount of starch? How is starch test conducted? So starch test is conducted using iodine. That we have discussed in last class, isn't it? Now we pluck a leaf from each pot, and what is the experiment we are going to do? We will dip the leaf in the dilute iodine. Now we will observe for change of color. Do the both leaves show the same observation? No. Why? Because the pot X, it has potassium hydroxide. It has potassium hydroxide, and this potassium hydroxide, this absorbs carbon dioxide. Whatever it is present here, it absorbs the carbon dioxide. So starch formation is not possible. While this, this leaf turns into dark blue in color. That means starch is prepared here. Starch is prepared here utilizing carbon dioxide. Got it? This shows the importance of carbon dioxide for photosynthesis. So, in the first jar, potassium hydroxide, it absorbs carbon dioxide. As there is no availability of carbon dioxide, starch cannot be prepared in the first jar. While in the second jar, the leaves will have more amount of starch. This can be done through iodine test. Okay, what is the conclusion? You should write the conclusion. What can be concluded from this experiment? Write the conclusion, underline that, so that examiner will understand that you have understood the experiment. Well, and you will fetch more marks. The amount of carbon dioxide affects the process of photosynthesis. What is the outcome of photosynthesis? Formation of carbohydrates. Here it is starch, isn't it? Because the carbon dioxide is absorbed by potassium hydroxide. Limited carbon dioxide or no carbon dioxide is available. Limited or no carbon dioxide is available here. So starch preparation is not possible here. While here carbon dioxide is available, so starch preparation is possible here. Got it? This is the experiment. This experiments can be asked for 8 mark questions. Now coming to the next raw material or element, it is water. How is water absorbed or how is water transported to different parts of the plant? If you observe here, this figure, it is in your textbook. If you observe here, the roots have hair-like structures. These are called root hairs. The roots have hair-like hair-like structures. These are called root hairs. These will help in absorbing more amount of water, and that water, it is conducted to different parts of the plant through xylem. The loss of water from the leaves, it is called transpiration. Okay, is water required for photosynthesis? Yes, water is split into hydrogen ions and oxygen. Oxygen is released as a byproduct, and that hydrogen ion is added. That is, the reduction happens, isn't it? And carbohydrates are formed. Root hairs provide large surface area to speed up the water absorption, and this is passed to different parts of the plant through xylem, Coming to carbohydrate preparation is done. What about proteins? Yes, do they require? As we require to build up, to grow, we require proteins. Even plants require proteins. They get in the form of nitrogen, and also they require other nutrients like phosphorus, iron, magnesium. They get it from the soil. Plants take up nitrogen from the soil in inorganic salts as nitrates or nitrites. As organic compounds, they will take up, that is produced by bacteria. Some leguminous plants, they have some bacteria which will help, these are called nodules, which will help in fixation of nitrogen, atmospheric nitrogen. It is absorbed by this bacteria and this is given to the plants. Okay, this is rhizobium bacteria. These are leguminous plants. You can find nodules over here. So the atmospheric nitrogen is taken up by this bacteria and this is given to plants. Okay. So plants prepare, through this we came to conclusion that plants prepare food, they absorb autotrophic type of nutrition and they prepare food through photosynthesis. Yes. Do all the plants prepare food? Yes, whichever plants are green in color, whichever plants have green pigment, that is chlorophyll, they prepare food. Okay. Coming to few are there which depend on other minerals or other nutrients on other organisms. For example, insectivorous plants, they photosynthesize because they are green in color, they photosynthesize. Yes, they do that, but they depend on for other nutrients, they depend on the insects which they trap in. For example, Venus flytrap, it depends upon the insects. It gets its nutrients from the insects which it traps in. Okay. This is about autotrophic nutrition. Hope you have understood this well. Next, coming to heterotrophic nutrition. Heterotrophic nutrition, there are many strategies to describe this heterotrophic nutrition, because in different organisms they perform different modes of nutrition based on different methods they use. For example, it may be saprophytic, it may be parasitic, it may be holozoic. Saprophytic, especially the decomposers. What is happening? These organisms, they break up the compounds into simpler one outside the body. Mostly these are tiny organisms. They break up this complex substances or compounds into simpler one outside the body. These are scavengers, or what you can say, decomposers. Examples, they feed, mostly saprophytic, they feed on dead and decayed substances. Example, you can see bread mold, you know. When you keep bread for so many days, put some water, you can observe that. That is an experiment you have done in childhood days. Put some water and keep it aside for 2 days. You can observe some greenish material that is nothing but fungus, that is bread mold. You know, mushrooms and yeast, all are the examples which perform saprophytic nutrition. In this, the saprophytes break down and convert the complex molecules into simpler ones outside the body. Got it? Then parasitic, parasites which depend on, first of all, they depend on other organisms, and they, in return, they don't give anything. But aim is the benefit kaadu, harm. They'll harm that host or organism. Those are parasites. Like this is louse, plural is lice, louse, tapeworm, okay, leeches, ticks are the examples which perform this parasitic nutrition. Coming to holozoic nutrition, these organisms, they take up the complex food inside the body. They take up the complex food inside the body, and they break down these complex foods into simpler ones, and from that they get energy. That energy is transported to different parts of the body to perform various functions. Holozoic nutrition, we perform holozoic nutrition. Example, human beings, example, man, amoeba, etc. Okay. On the basis of feeding habits, the organisms, on the basis of how they feed, how they depend on other organisms, about that. Even in your topic like environment, you get that these are first are producers, next are primary consumers, you can say those are herbivores, next are secondary or tertiary consumers, you have carnivores, you have omnivores, you have this is classification based on the feeding habits. Now, coming to the nutrition in animals, nutrition in plants, we have seen all the green plants, they perform autotrophic. Exception, you have seen some, they depend on other organisms like insectivorous plants. Though they photosynthesize, they depend on other organisms like insects. They get that nutrients from the insects that they have trapped in. Now, coming to the nutrition in animals, generally this is divided into 5 steps. The sequence of these 5 steps, you don't miss the sequence. Okay. First is ingestion. First process, what happens? Yes, better. First process, what happens? Food manaki lastundi. Okay, by environment nundi techukuntunnam annam. Our food, we should intake it, isn't it? Edanna undali. Manaku aithe specialized organs unnai. Ade unicellular organism kaithe anni okate chestadi. One cell will do everything. Ingestion, intake of food. Next, it is digestion. Whatever it is taken in, that should be broken into simpler forms. That is digestion. Later, it is absorbed. That should be absorbed into bloodstream or anything. Next, it is assimilation. Absorb ayinadi energy ivvali. Enduku absorb chestunam? Vere cell organelles ki, vere tissues ki adi energy ivvali. Appude kada perform chesedi vere functions. It should perform all tissues and cells, they should perform various functions, isn't it? So assimilation in a process to provide energy. Next is egestion. Egestion ante undigested food edaithe undo that is discarded outside the body. This is the sequence. Don't miss the sequence. In any animal, this is the sequence. Ingestion, digestion, absorption, assimilation, and fifth one is egestion. Coming to amoeba, look at the picture. This diagram of amoeba is important. This can be asked as a picture one, picture based question, or asked as a 8-mark question, including this nutrition in unicellular organism. You can choose amoeba. Amoeba is unicellular organism. Yes, it feeds on phytoplanktons and zooplanktons. Phytoplanktons ante microscopic plants or animals that are floating in the water. It feeds on them. Okay. First figure ela istaru, amoeba irregular shape. ఎలాగైనా వేసుకోవచ్చు, ఇర్రెగ్యులర్ షేప్ కాబట్టి మీరు ఎలాగైనా వేసుకోవచ్చు. అమీబా షేప్ వేసిన తర్వాత ఫస్ట్ ప్రాసెస్ ఇంజెక్షన్. ఫస్ట్ అమీబా ఏం చేస్తుంది, ఇట్ షుడ్ ఐడెంటిఫై ద ఫుడ్. First it will identify the food. After that it will produce finger-like projections. It will produce finger-like projections. Those are called pseudopodia. Amoeba figure elagaina veyandi. Idi nucleus, idi cytoplasm. This is amoeba. Okay. Next amoeba vesin tarvata these are finger-like projections. Ila finger-like projections veyandi. Okay, finger-like projections. These are called pseudopodia. Pseudo ante false. Pseudo ante false. Podia ante limbs or feet. Podia ante plural, podium ante singular. Pseudopodia. Now it has identified this is a food particle. Every time draw the nucleus. This is a food particle. It has identified. Now it has produced pseudopodia. Next what will happen? Next figure. A vacuole is created. A vacuole is created. Vacuole is created and this will engulf. It is engulfing ante catching it. It is engulfing, catching it, the food. It will engulf the food. Next what is happening? This food enters inside. This food is entering inside and what is happening? This food is broken down. That is digestion. It is broken down and it is absorbed here. Next figure, absorption. Ikkada absorption and rasi. Now here you don't find any food vacuole. Food vacuole disappears. Food vacuole disappears. Now coming to the shape, this cell membrane is ruptured so that the unwanted or undigested food is discarded out. The undigested food is discarded out. This is egestion. This is egestion. Okay. This is the process of nutrition in unicellular organism amoeba. Ikkada chinnaga vesi run growth chuichandi. Koncham peddaga veyandi. Growth chuichandi. With this I am unable to do so. It's not fine. Neat ga veyandi. Got it? So you can see, look at the picture. You have a neat picture in your textbook. You can see that. So what happens? First is ingestion. It develops pseudopodia and vacuole is created, which helps in what is happening, which engulfs the food. This engulfs the food and enzymes enter this vacuole so that it is digested. During absorption, the vacuole is disappear. Absorption, vacuole disappears, and the digested food, it enters into the cytoplasm. Yes, now amoeba is growing, assimilation, amoeba is growing. Now undigested food is thrown out, that is digestion. Okay. Now, coming to assimilation is the process of uptake of substances or nutrients by tissues for their use. I explained this. Next, coming to the next unicellular organism, that is paramecium. Look at that shape, it is like slipper, right? Look at that. Contrary to amoeba, or unlike amoeba, amoeba has only single cell. It is single cell, isn't it? Everything is performed only by that single cell. Simple process diffusion, through simple process diffusion. Here it has one opening for intake and one opening for egestion. One, it is called cystome, that is cell mouth, and for egestion it is called anal pore. Two it has. That differentiation you should know. And these are the food vacuoles. So what is happening in paramecium? Food is brought to cytostome through the continuous beating of, you can find hair-like structures around the outer surface of paramecium. Those are called cilia. Food is brought to cytostome through continuous beating of this hair-like structure. They continuously beat and they bring the food through to the mouth, that is cytostome, and from there it enters, and the rest of the process it continues. As I have explained, like enzymes are released, digestion occurs. Digestion means enzymes are released. In the first class I told you, complex substances are broken into simpler one through biocatalysts called enzymes. Okay, enzymes are released, so that is broken into simpler substances, that is absorbed, and energy which is released, that energy is utilized for the entire cell, that is called assimilation. Undigested food, it is discarded through anal pore. Okay. Then food is digested in food vacuole, that I told you. Next, coming to nutrition in human beings, this is through the process digestion. I will explain the entire one through this figure. Details, you can take a screenshot. I have given a tabular column. You can take a screenshot for that. Entire digestion I am going to explain with the help of this diagram. Can I zoom in? Okay. First process is ingestion, intake of food. Intake of food is through mouth. That is called oral cavity or buccal cavity. Inside the mouth, what do you have? You have tongue, you have teeth, you have salivary glands, isn't it? Tongue, teeth. Teeth are used to break down this complex food, whatever we take, into simpler. We cut here it is cutting, isn't it? You know, the teeth, first incisors. We cut it. You cut it into in small pieces. Tongue is used to mix up this food well. Three salivary glands are present. They are parotid gland, sublingual gland, and submandibular gland. These salivary glands, they produce saliva. What is the function of saliva? Saliva, it adds some lubrication to the food, and also it contains an enzyme called salivary amylase. It contains an enzyme called salivary amylase. This salivary amylase, it breaks down carbohydrates. Here it is starch. Right? Here it is starch into simpler molecules. Before going to that, I want to tell you about this carbohydrates, the carbohydrates are basically of three types. Those are monosaccharides. I'm writing in shortcut, children. Monosaccharide, disaccharides, and polysaccharides. Monosaccharides, examples of this are glucose, fructose, galactose. Disaccharides, mono and a single sugar. The glucose is the simplest form. These are the simplest forms of sugars. All these are sugars, simplest form. C6H12O6. Simplest form. Ante carbohydrates, complex substances, they break down into these simplest forms. Disaccharides and two sugar molecules, those are called disaccharides. Maltose. Maltose has two glucose molecules. Maltose has two glucose molecules. Lactose, you have this in milk, isn't it? It has one glucose and one galactose. One glucose and one galactose molecule. Then sucrose, in sugar, you have common sugar, table sugar we eat, isn't it? Sucrose, that is sucrose. It has one glucose and one fructose molecule. Coming to polysaccharides, starch, and many sugar molecules, glucose molecules, n number of times తీసుకుంటే అది starch, poly అంటే many. Then glycogen, cellulose. Okay, remember this cellulose, we'll discuss about this. Glycogen, I told you in plants, the undigested, the excess of sugar or carbohydrates, it is stored as starch. In human beings, it is stored as glycogen. So here, this starch is broken down and converted into maltose. Okay, next, it enters pharynx. Pharynx, you know, pharynx, it is what you can say, it is between respiratory and this digestive system. That is a common junction you can consider. Okay, through pharynx it enters. While the food particles don't enter, they should not enter the lungs, isn't it? There is a protection for that, that is epiglottis. It is like a lid. It is like a lid which closes, the aperture like thing is present. I'll show that when I discuss respiration. That is, this epiglottis closes the glottis so that food doesn't enter larynx and to lungs further. So from pharynx it enters into esophagus. Esophagus is a muscular tube-like structure. It is a tube. It is called food pipe. It is called food pipe. It's a tube, muscular this thing. And food enters here through wavy-like motion. It is not like this. The food enters through wavy motion. This is called, muscle contracts and next it relaxes. When it relaxes, food enters, and it contracts, it pushes. Like that, this is called peristalsis. Even here, the peristaltic movements occurs even in the intestines too. So this is here through esophagus, the food enters through esophagus and through peristaltic movement, and it enters the stomach. It enters the stomach. Stomach is lined by three tubular gastric glands which secrete gastric juice, and it has mucus. Why mucus? What is the function of mucus? As I told you, mucus adds lubrication, and also as HCl is present here. HCl is, what is the pH of HCl? 0 to 1.1. So it is acidic. It is acidic. As HCl is present here, the inner lining of the stomach may be eroded. Acid veste emaitadi beta kachitanga kalpotundi, it will be burnt. To protect that inner lining of this stomach, it is lined by this mucus, prevents this erosion of this inner lining of stomach, which can be by HCl. So the gastric glands here, they produce gastric juice. The gastric juice, they contain enzymes which will break down the whatever the bolus, this is called bolus, when it enters here to here, this is called bolus. This will break down into simpler compounds or molecules, okay, through enzymes. What are the pepsin? We have seen till here, we have seen carbohydrates are broken down. Now pepsin, this acts only in acidic medium. So the presence of HCl is required. Pepsin, it breaks down proteins into simpler one. Now here you can find, this is the first, you can find the junction here, it's a J-shaped one. You can find here, this is the first part of intestine, and that is called duodenum. This first part of this intestine, that is called duodenum. This duodenum, it will receive the food that comes from stomach. It will receive the food that comes from stomach. It is basically acidic because HCl is acting on it, isn't it? That acidic need to be converted into alkaline. That can be done through bile juice. You can see a green part here, that is called gallbladder. Liver produces bile juice. This bile, it helps in fat emulsification, that is breaking down of fats into simpler compounds, simpler ones. Bile is stored in this gallbladder. Okay, that bile enters the duodenum, and you can observe the pancreas over here. Pancreas, that will secrete pancreatic juice, which contains enzymes. You have seen salivary amylase over in the mouth. Here you have pancreatic amylase, which will break down the carbohydrates further. Amylases break down the carbohydrates. Next, it is trypsin, other enzyme. I told you enzymes are very essential because they'll act as biocatalysts. They'll break down complex substances into simpler substances. And lipase, the term itself says, ase is the suffix added to enzymes. For example, to break down the lipids, it is lipase. You have seen ATP synthase. Ase is added like that. Amylase. Lipids are broken down, that is fats are broken down into simpler one. So this pancreatic juice has all these enzymes. They enter duodenum. Then intestinal juice, because duodenum is the first part of intestine, right? Intestinal juice, this contains enzymes which convert carbohydrates into simpler, what is the final one? Yes, glucose. Simplest form of carbohydrates is glucose. Next, proteins to amino acids. The simplest form of protein, it is amino acid. Fats to fatty acids and glycerol. That is the final one of fats. Now, as the food is broken down into simpler ones, did you understand better? It is broken down into simpler ones. It is now absorbed. It is now absorbed in jejunum and ileum. These are the next two parts of small intestine. Okay, these are the next two parts of small intestine. So, the overall length of the intestinal tract is approximately 27 feet. It is 27 feet. It is 8.5 meters. One thing it is, you need to know, that question was asked in CBSE examinations too. Which of the organisms have longest small intestine? Yes, herbivores have longest small intestine. Why? Because they consume grass, isn't it? Grass contains cellulose. Cellulose should be broken by an enzyme called cellulase. This breaking up of cellulose is the toughest part. So the cellulose that is kept in the small intestine for longer time, so that it is infested by the bacteria too. Bacteria act on it and break down that into simpler molecules, simpler forms. Okay, so herbivores, that is the animals which are grass eating, they have longest small intestine. Not in carnivores. Carnivores have shorter. In comparison with the herbivores, carnivores have, because meat absorption is easy when compared to the grass absorption, the breaking down of the grass. We can't digest grass because we don't have the enzyme cellulase to break down cellulose. Cellulase is required, which is not present in human body. Andike manam gaddi digest chesukolam, gaddi tinalem. Got it? Okay. So when food is released into the first segment, that is duodenum, this is controlled by a ring of muscles. Those are called sphincters, children. Those are called sphincters. Only for your knowledge I am giving that. Those are called sphincters. Now, for the absorption, to increase more absorption, finger-like projections, finger-like structures are present in the small intestine. Those are called villi. Those are called villi. They will increase the area of absorption and also the rate of absorption. Now, the undigested food, whatever it is present, the undigested food, it enters these are the parts of, so this is too sensitive. This is small intestine, right? The red one you can observe, this is large intestine. After absorption, the food enters into this large intestine. Absorption and assimilation, everything happens. That is, energy is given off to the different parts. Through blood it is carried to different parts of the body. Now the undigested, unwanted food, it enters into this first part. This is cecum. Here you have vermiform appendix. Now food enters into this first part of large intestine, it is cecum. Here you have vermiform appendix. It is vestigial, it is a vestigial organ. Vermiform appendix is a vestigial organ. That is, it's of no use. When some food enters into that, it will rupture, so it need to be removed. That is through appendicectomy, that is removed from the body. Okay, so when undigested food, it enters into the first part of the large intestine, cecum, and from there it goes to this is ascending colon, this is transverse colon, and this is descending colon. Here still the undigested food is further broken down. Some of the bacteria are present over there and water is absorbed here, and that is discarded through rectum and finally anus. Okay, this completes the topic digestion. So what are all the organs involved in digestion? First it is oral cavity or buccal cavity, and next it is esophagus, and from there it is stomach to small intestine. Small intestine has three parts: duodenum, jejunum, ileum, and from there to large intestine. Absorption, it takes place in jejunum and ileum, and digestion, it starts from the mouth to duodenum. Then that food, whatever it is absorbed, that later on process, energy is released and it is taken up through blood to different parts of the body so that they can perform different functions. I think it is clear. I have provided everything in a tabular column. If you want this, you comment below. I'll provide this so that you can understand this easily and you can make notes out of this. children. This is very easy. You need not study the entire topic of digestion in your textbook. Just to go through this tabular column, you can make notes and you can definitely fetch more marks. Okay? All what are the enzymes secreted, everything I have given. Okay. In the next topic, I am going to cover respiration, transportation, and important questions if possible. As this is a vast topic, I am discussing everything in detail better. And if you want, as I told you earlier, if you want anything, if you think any topics in physical sciences, if you consider them tough, please comment below so that I can make a video on that too.